Hello my churku people, I welcome you all to score 360 out of 360 in NEET Bio within 250 days, and today we are going to talk about the third topic of our chapter, that is molecular basis of inheritance. Topic is RNA versus DNA as genetic material. So today we are going to see a fight, and also the most famous experiment, that is Meselson-Stahl experiment. But what did we cover previously? We spoke about nucleosome, the structure of it, and also the experiments which ran behind the search for DNA. How did it look, or was it the genetic material or not, and stuff like that. So we covered all of it, and we have now reached this point. And I also want to tell the number of questions was 6 the previous year. Before that it was 10, again 6, again 9. So this year we can expect that it could shoot up and could give you more questions as well. So be prepared, children. Please don't lose your 40 marks, okay, or don't lose your 24 marks. Either way, you don't have to lose even a bit of your marks if you study with us properly. Now, were you able to solve this question? Let's see. What are the structures called that give an appearance as beads on a string in chromosomes when viewed under electron microscope? So there are certain structures like this onto which the DNA, or poor DNA, wraps around, right? What do you call this? And it looks like beads on a ring, right? So what are these structures, children? These are none other than nucleosomes. Okay, they are none other than nucleosomes. So children who were with me in the previous session know it definitely, and who are new, it's okay, yeah, we can still study it, yeah. Now let's start today's topic, yeah. So DNA versus RNA as a genetic material. We will read a few things and then we'll go ahead. I'll explain you each and every point, okay. So it became an established fact that it is DNA that acts as a genetic material. We proved it with the earlier experiments, right? However, it subsequently became clear that in some viruses, RNA is the genetic material, and you have to remember the name of them. For example, tobacco mosaic virus, Q beta bacteriophage, etc. So will you remember the name? It is, first one is tobacco mosaic virus and Q beta bacteriophage. So this bacteriophage is different from the bacteriophage we studied in the other experiment, okay. So over here, both of them are having RNA as their genetic material instead of having DNA as their genetic material, and it's given in your NCERT, which is why I am making you write this, okay. Now answer to some of the questions such as why DNA is predominant genetic material whereas RNA performs dynamic functions of messenger and adapter has to be found from the differences between the chemical structures of the two nucleic acid molecules. So with this information, they are trying to tell you that DNA has always been acting as a good genetic material, whereas RNA, even though it's a genetic material, but it is more renowned for what? For acting as what? A messenger. For acting as a messenger of what proteins has to be made. Remember the central dogma, DNA converts to RNA, not converts, DNA helps in the formation of RNA and RNA helps in the formation of proteins. So basically the message that has been passed is passed by the RNA. So RNA acts as messengers as well as what? Adapters. We'll come to know what do you mean by that. But so the role of genetic material has always been assigned to DNA, whereas the role of a messenger and everything that comes to play has been assigned to RNA because of their structural differences, children. Okay, so this is given from here to here. Now something that is very important which has come in the previous year questions. Listen to me carefully. A molecule that can act as genetic material must fulfill the following criteria, and it's a direct question. What are those things or what are the characteristics needed to be a genetic material? Okay, this has come directly like that. I can show you a question as well. Okay, first point, it should be able to generate its replica. Okay, so that means over here, if I talk about DNA, and on that side if I talk about RNA, and if you talk about replication, what do you mean by replication? So what happens is that, suppose this is a DNA strand, okay, A is here, G is here, C is here, G is here, T is here, A is here, and things like that. So while replication is happening, replication means you are making a copy of something. So this particular strand which is coiled like this, it uncoils itself like this, and later certain enzymes break them down into 2 strands. Okay, over here what all did we have? We have A, we had C, we had T. Over here what all did we have? We had G, we had G again, we had A. Sorry, not G here. Oh my god, how can I do that? Okay, it was T. Here also I need to change. Okay, here it's T. Right? So I just uncoiled, I opened it and I break the bonds which were here. So I broke the bonds which were here. Now, what happens? I need to make a replica of it. So I make a replica of it. A will be pairing with T, C pairing with G, T will be pairing with A. If you have noticed, I have made a strand like this. Similarly over here also, A, G opposite C, A opposite T. Now I have 2 strands like this, which is a replica of the DNA. Okay, and then these guys will come back to the same position again. They will coil themselves again. This is known as replication, where you are copying the same thing, you are making more copies of yourself. Okay, so DNA has to be capable of replication, because why it has to be capable of replication, or any genetic material has to be capable of replication because it has to be passed down to the next generation. For example, my father has his own set of DNA. Now, can he give his own set of DNA to me? No, children. He will give a replica of his DNA to me. So that is a use of replication. So whenever the cell is dividing, it is giving its replica. The DNA is replicating itself and giving that replica to the next cell. Okay. Now, so replication is a very important process. So to be a genetic material, you have to show replication. DNA shows replication. What about RNA, children? RNA also shows replication. So the first point is absolutely clear, I hope. So replication. Okay, we'll do one thing. I'll write it over here. So if we talk about replication, this is DNA and this is RNA. Both of them, they do it. So the first criteria, I cannot decide that who is the better genetic material. Second criteria is it should be stable chemically and structurally. So I am writing stability. Now, if you were with me from the start, you might be knowing one very important thing. That is, RNA has always a history of being less stable. So in the case of RNA, instead of having T, they do not have T, instead they have U. U stands for uracil. And uracil is basically reactive. Uracil is basically reactive. It's not something that is stable. That is one thing. Okay, they have a different pyrimidine. That is one reason that it's not stable. Second reason for it to be not stable is that if you have seen the sugar, you know they have ribose sugar, right? So 1, 2, 3, 4 and 5th carbon is over here. So in the case of deoxyribonucleic acid, that is DNA, they do not have an OH group on the 2nd carbon. They do not have this OH group on the 2nd carbon, instead they just have H. H is fine. But when OH comes, this OH group is reactive again. And this reactive OH group will be a problem, because if it goes on reacting with certain other stuffs, obviously it is going to damage itself in the process, right? So T2U, that is uracil being there, is a problem. Also on the 2nd carbon, please remember, on the 2nd carbon of ribose sugar, what do we have, children? We have OH group, alcohol group, which is reactive, and we cannot afford that. But on the case of DNA, it is said that RNA came first and then came DNA. So what had happened? The mistakes are mixed. The mistakes, basically whatever mistakes were there, it was corrected. Okay. So in them there is deoxyribose sugar. Okay, 3, 2, 1. That means here there is no OH group, instead it's just H. So it's all safe. Second thing, uracil basically was replaced by thymine, and thymine is what? Methyl uracil, 5-methyl uracil, as in it is now having a methyl group. So the pre-existing uracil had a methyl group on it, and now it has formed thymine, which is more stable form of it. It's not reactive. Okay. So if I talk about stability, this is stable, this is not stable. Okay. So it should provide the scope for slow changes that are required for evolution. So yes, there are chances that the DNA strand which is there, it might have some mutations in itself. Okay, mutations, do you understand? We studied in principles of inheritance and variation, whereas one small change in the base, for example, C has changed to something else unknowingly, okay, because of a mistake that has happened. But this mistake is not costing me my life, okay, but it is responsible for some amount of variation. So usually what happens is that you have a DNA, children, and the DNA, basically you have codes like this, right? So each code codes for, and this is a codon, okay, it codes for a amino acid. Similarly, you will have next codons also, they will also code for amino acid. These amino acids basically come together to form what? The proteins. Now, if one code changes, obviously the whole thing, the codon whole in total will be changing, and it will be different amino acid making a different protein. This is how we have variations in our body. Okay, some variations, some mutations can lead to death, can cause diseases, whereas some mutations is okay. It's okay to have them. Okay, so mutations are totally fine. They have it. DNA has it. Okay, so I am writing over here the third point, is changes in genetic material. Okay, so over here they have changes, slow changes. Okay, I am writing slow changes. That is good. But on the other hand, when you talk about RNA, you saw how unstable this is. So it will be rapid changes. And rapid changes, children, it can lead to dangerous outcomes as well, which we do not want. So this change, a lot of things change, different amino acids came, different proteins came, which is harmful for us, or probably a functioning of one particular thing has stopped because of this change. So this is a problem. So RNA has rapid changes because it has a very unstable structure. On the other hand, when you talk about DNA, it has slow changes, which is comparatively fine. Okay, we are not rapidly changing around. So this point is done. Last point. It should be able to express itself in the form of Mendelian characters. That means basically the DNA which is there, okay, the DNA which is there, it should give out a character. So how does it do? DNA basically gets converted to it helps in the formation of RNA, which RNA? mRNA, messenger RNA. This process is known as transcription. We studied about it, right? Now mRNA is basically translated to what, children? Protein. mRNA is translated to something that is known as protein. Okay, so this protein will be giving a character. This protein, for example, this is a protein that is melanin. Melanin gives the color of the skin. Okay, there is one other protein, keratin. Keratin helps the formation of hair. So this is the character that one is having, right? So basically, you have DNA, mRNA, protein, and character. So yes, it shows Mendelian characters. It's all good. Talking about RNA, RNA directly can help in the formation of protein. They do not need to do transcription, right? Transcription is when DNA is forming RNA. Over here, it's easy for them, so they are also doing it, and it will be really fast as well, okay, compared to them. So with this conclusion, with this particular thing in mind, okay, over here I didn't write what is the, okay, whether they are responsible for characters. Yes, children, they are responsible for characters. They are also responsible for characters, but somehow, if you see the score, RNA is kind of losing. Why? Because it has a very unstable structure, which is why it can lead to rapid amount of mutation, and if you go like that, probably we will die. So we don't want RNA as a genetic material, we want DNA as a genetic material, and that makes us unique. Okay, remember? Done? So these are the things that you need to remember. PYQs have come directly from this topic, so I want you to remember it very clearly. Okay, and please remember tobacco mosaic virus as well as Q beta bacteriophage, the other bacteriophage that we studied in the experiment. No, what was the name of the experiment? I am not able to recollect it somehow. The E. coli experiment. Somehow I am not able to remember. I remember the year as well, 1952, but somehow the name I forgot. Hershey and Chase, yes, it is a Hershey and Chase experiment. In that, the bacteriophage was different. This is different, okay. This is Q beta bacteriophage, which has RNA as a genetic material. So Proven that DNA is most good amount of genetic material, whereas when you talk about RNA, it acts as a messenger, adapter, protein, stuff like that. Okay. Now going ahead, what was the first genetic material? So RNA is saying, at least give me this title. I want to be the first genetic material. So yes, I have given the title. RNA is the first genetic material, children. RNA is the first genetic material, and as I said, it had uracil, it had ribose sugar, okay, which made it less stable, which is why we got DNA which had thymine in the place of uracil and deoxyribose in the place of ribose sugar. Works? Is it okay? Now, why was RNA initially doing really well? Because it was acting as a genetic material. Because it was acting as a genetic material, as well as one more very important thing is that they were also acting as catalyst. As in, usually, you know what? For something to convert from this to this, you need the help of enzymes. And these enzymes are, children, proteins. Okay, but suppose we do not have proteins, how can we make the enzymes? So basically over here, RNA, which was genetic material, was acting like a catalyst. Okay, it was itself acting like an enzyme which was helping in the conversion of this to this. And the example of one such catalyst is ribozyme. It is made up of RNA. Ribozyme, it's an example of catalyst, RNA catalyst. So we do not need proteins for the conversion, instead we had this. So there were two reasons for its success initially, and now later on when we saw that human organisms are getting complex, now there are human beings as well. So in human beings, RNA cannot be just the genetic material over there. Even though we have RNA in our body, it's not the genetic material. Why? Because we need to have a much better and a safer way to survive, and that is why all of this happened. So with evolution, we got DNA which had these changes as well. Cool? Works? Now, RNA was the first genetic material. There is now enough evidence to suggest that essential life processes such as metabolism, translation, splicing as in cutting and things like that, you will get to know about it later, evolved around RNA. RNA used to act as a genetic material as well as catalyst. There are some important biochemical reactions in the living system that are catalyzed by RNA catalyst and not by protein enzymes. Yeah, and the example, one such example is what? Ribozyme, children. Please remember. But RNA being a catalyst was reactive and hence unstable. So it being a catalyst is obviously a problem, right? It is reactive, it is able to do the reactions, right? That is also a problem, and it's reacting because of its structures as well. Now, therefore it is unstable, and hence DNA has been evolved from RNA with chemical modification that make it more stable. So what is more stable, children? DNA obviously is more stable, I told you. Fine. DNA being double-stranded and having a complementary strand further resists changes by evolving a process of repair. So we are, in our body, we have a wonderful DNA repair mechanism, which is why even when I am talking, there are 10,000 mutations probably happening in my body. There are 10,000 mistakes that are happening in my body, which is being repaired by our own sets of things, our own set of protein which is there in our body. Okay, when they fail, that's when we have certain diseases. But we have DNA repair mechanism, and with this everything got better as well. Okay, so they are giving credits to DNA complementarity as in the double strand, which makes it more stable. When you talk about RNA, RNA, children, is a single stranded or double stranded, children? It can be single stranded, it can be double stranded as well, but most of the times it is single stranded. Okay, so it can be single stranded or it can be double stranded. And there's an important question that I remember from this. RNA does not follow the Chargaff rule. What does the Chargaff rule say? Remember, when A is there, A will be having enough amount, same as T. And if G is there, there will be enough amount of C as well, the same amount. For example, 20% this is there, then 20% this will also be there. 30% this is there, then 30% this is also there. But in the case of RNA, if you think it will be single stranded, can you expect this to be same? No. A, T, G, C, it can be anything, right? It does not have a complementary pair. When it has a complementary pair, I can say that, okay, if this is here, the complementary pair will be a pyramid and it will be equal. But over here, if this is single strand, I cannot think I will follow this. So I can say RNA single stranded RNA do not follow Chargaff rule. Works? Okay. Now, going ahead, the semi-conservative DNA replication, and this is a topic that I love so much. So, we will be reading about it. So, let's read it first and then we will go ahead. While proposing the double helical structure, that is this is one helix, this is another one, okay? Helical structure for DNA, Watson and Crick had immediately proposed a scheme for replication of DNA as well. To quote their original statement, that is as follows. So, they said this: it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material as well. In the year Watson, in the year 1953, Watson and Crick said this, okay? So, the scheme suggested that the two strands would separate and act as a template for the synthesis of new complementary strands. After the completion of replication, each DNA molecule would have one parental and one newly synthesized strand. This scheme was termed as semi-conservative DNA application. For your understanding, let me explain it to you. So, for that we need to make a proper diagram. Cool? Something that I explained earlier, but yeah. A, then here T, if here G, then here C. If here C, then here G, again T here, so A here, okay? Remember adenine and thymine and guanine and cytosine, right? Now C here, then it's G, and T here, again it's A. If A here, then it's T, okay? This is one strand, children, and this is the second strand. Both of them, they are parental strands. This is parental DNA, children. Now, I want to make a copy. So, for that, what I will do? The first thing that I will do is unwinding of DNA. I will try to open the helix, and now it will appear to look like this. I am making it in a distance so that I can do everything here itself. Okay, I should be making in much more distance. Okay. Fine. Over here I had A, G, C, T, G, C, T, A. Here I had T, C, G, A, C, G, A, T. Right? So they unwind it, and now depending on what base pairs are over here, I'll make a new strand, new complementary strand. So here A is there, so I'll bring T over here. Here G is there, so I'll bring C over here. Here it will be G, here it will be A, here it will be C, here it will be G, it will be A, here it will be T, and then tada! I have a new strand being formed. This is what, children? This is a complementary strand. This is what? A complementary strand. Okay. Similarly, over here also I will have a strand. A, G, C, T, G, C, T, and A. Did you get it? Over here, what is happening? Are you able to understand this? And at the end, what are we getting, children? At the end, what are we getting? We are getting, okay, each molecule would have one parental and one newly synthesized strand. So over here, at the end, I will be getting something like this, where there will be one parental, which is in yellow, and the other one is a newly synthesized strand, which is why it is semi-conservative. So I'm not joining them back. Okay, it's what basically I am conserving one parental DNA over here. So this is what? Semi-conservative. This is parental, and this is the newly synthesized one. This is parental, this is newly synthesized one. Together basically what are they forming, children? This is the semi-conservative model. Okay, this is what the semi-conservative model, because I am still having one parental thing. Cool, works. So this was again suggested by whom? Children, our own Watson and Crick in the year 1953. So over here I made, just understand, okay, I'll take a slight change over here. For your understanding, I'll make like this. Okay, works? Fine. So that you don't say that, oh ma'am, there she did something else and here she is doing something else. Basically the same thing itself. I wanted to show that replication is happening there. Here we would have anyways spoken it in a bit detail, right? So this is also done, children. So we studied DNA versus RNA and RNA's world, that is RNA is the first genetic material, why it is first genetic material, and the semi-conservative mode of DNA replication as well. Now we'll come on to the most famous Meselson-Stahl experiment to prove the semi-conservative replication. Obviously we are dealing with biology and biology has experiments to prove things, otherwise you cannot believe it, right? Obviously, for obvious reasons. So according to Meselson and Stahl experiment, what did they do? Let's see that. So it was Matthew Meselson and Franklin Stahl who performed the following experiment in the year 1958. Okay, so 1952 the Hershey and Chase did their experiment of proving that DNA is the genetic material. Over here 1958, almost 6 years afterwards, Meselson and Stahl did their experiment to prove the semi-conservative replication as well. So what did they do? They grew E. coli in a medium containing NH4Cl. Okay, and here N15 is a heavy isotope of nitrogen. It's a high isotope of nitrogen, it's not radioactive. Okay, so you have different isotopes, right? N14 is there, N15 is there. So here they are growing what? E. coli. So E. coli is a bacteria, and this E. coli is being grown in a medium that is containing N15 NH4Cl. Okay, here N15 is there, please remember that. Okay, and N nitrogen is a part of DNA, that's why I am very much specifying this. Okay, so he grew this, and then the result was that N15 was incorporated into newly synthesized DNA as well as the other nitrogen containing compounds. So he found that when he grew the E. coli in this particular medium, basically the DNA had this incorporated. So the DNA, I can write the DNA which is there, it has N15. Okay, both of them, they had N15 variety of them. Now this heavy DNA molecule could be distinguished, okay, this DNA molecule could be distinguished from the normal DNA by centrifugation in a cesium chloride density gradient. And please note that N15 is not a radioactive isotope and it can be separated from N14 only based on the densities. So it's not something radioactive, okay, that you can see and then understand that it can be different from N14. No, it's not like that. Okay, it's not like that. We have to put it in something that is known as cesium chloride, and over here we will be finding that the ones which had 2 bands of N15, okay, this is the basic, I'm telling you the basic, which had 2 bands of N15, obviously it is heavy isotope, okay, so it will be dense, and it is a density gradient medium, that is of cesium chloride, so it will be coming towards the down. So I am writing this is N15 and N15. On the other hand, if it was something else, for example, there is a different variety of it that they have mentioned, and that is N14, okay. So suppose there is a DNA strand here where N14 has been incorporated, okay, both the places. So N14 is very light, so it will come towards this side. It's very light, so it will be towards here. So it was N14, N14. In the gradient chart, I am telling you all the gradients together, okay. Now there is a type where you have one strand as N15 and the other strand could be N14. In this case, what will happen? So it is a hybrid strand that is being formed. So I am writing N15, N14. Obviously, it won't be in deep because it is not having both of them as the same thing. It is not light also, so it comes in the middle. So this thing is for hybrid. Okay, so what he did was he grew E. coli in a medium that was containing NH4Cl, where N was having the heavy isotope, that is N15. Okay, he found that the DNA has incorporated N15. He did it for so many times such that the DNA has just now 2 strands of N15. Cool. And then he distinguished the DNA, that is different types of DNA, with the help of why centrifugation, where cesium chloride was a medium. Please remember, cesium chloride density gradient. Okay, it was a medium. Fine. Now, next thing that they did was, then they transferred the cells into a medium with normal NH4Cl and took samples at various definite time intervals as the cell multiplied, and extracted the DNA that remained as double-stranded helices. The various samples were separated independently on CSL gradient to measure the densities of DNA. So what happened? Now we have this thing, agree, because we were putting in this particular medium which had N15 as a isotope, right? So all of the DNA would had this particular strand. So have I not? Okay. Now, listen to me very carefully, okay? So, this strand is N15, this strand is also N15. Now, if I am putting the E. coli, just imagine that the E. coli is there around it, okay? E. coli in a medium now, which had 14 NH4Cl. Here, the isotope has changed. This is 14 now. So obviously, if I say, now again the DNA replication, the E. coli has to replicate itself, right? So DNA has to replicate it. So what will happen? There will be one strand over here. There will be one strand over here, okay, which is N15, or yeah, here also it will be 15N. Actually, you should write like this, not N15, okay? Please write like this, 15N over here also. But DNA replicates, so it changes itself, and now you have another complementary strand like this. But this is, since it is kept in a medium which has this isotope, it will be 14N. Here also it will be 14N. Okay. Now, if I do centrifugation, what will I find? I'll find that the DNA is there in the middle. The DNA is there in the middle, it's not in the bottom, it's in the middle because it is showing hybrid nature. Okay, that means probably the DNA has replicated and it has incorporated the N14 version of it over there, right? Now, still it's not done, okay, still it's not done. Imagine that the same experiment was repeated again. Okay, so it takes 20 minutes for E. coli to complete the cell division. So in first 20 minutes, okay, so in first 20 minutes, when he put it in this thing, okay, just a second. In first 20 minutes, he found that all of them were hybrid. Now, what happens in the next 20 minutes? Let's see that. In the next 20 minutes, I have a bigger box. So together it's actually 40 minutes. Okay, after 40 minutes of the initial start, I am getting all this thing. Let's see. From here I have started, okay, not from here, I have started from here. So initial 20 minutes, I got this where I had both of them. And then after that, next 20 minutes, I am going to see again what will happen. So I have 2 strands of normal DNA. Okay, when this strand will get separated and you have 2 strands like this, okay. Similarly, this strand will also get separated and you have 2 strands like this. Okay, they open up over here, and now I have to make a replica copy. But this time replica copy again I am putting in N14 isotope medium. Okay, so what will happen again? The DNA strand which will come here will be 14. So this is 14, but this is 15. Okay, what about this? Both of them will be 14. What about this? This will be again 15 and 14. What about this? This will be 14 and 14. Now this time when I did centrifugation, I found that 2 of them basically in the middle zone and 2 of them in the higher zone or the light zone. Okay. So over here, I found that it was N15, 14N, whereas over here I had N14, N14 on the light band. Okay, and you do this again and again repeatedly, you will be finding whatever you do after 20 minutes again you try to do, okay. After 20 minutes again you try to do the experiment. How many number of chromosomes will be formed? How many number of chromosomes will be found? Please remember, use this rule 2n to find the number of, not chromosomes, how many number of DNA will be formed. For finding that, use this formula, that is 2n. That is, in the case of in 80 minutes it is, okay, you are talking about 60 minutes, right. If it takes 20 minutes for the E. coli to replicate, okay, then in 60 minutes how many times it will replicate? 20 plus 20 plus 20. 20 plus 20 plus 20, 40 and 20, 60. So 3 times it will replicate. So 2 is to the power 3. Okay, 2 is to the power 3. What is 2 is to the power 3? 2 into 2, 4, 4 into 2, 8. So there will be 8 DNA. Okay, 8 DNA copies that will be formed, and out of 8 DNA copies, whatever you do, 2 of them will be coming in this format. Okay, 2 of them, because there is one strand over here. So again when it opens up, it will be looking like this here. Okay, and then a green strand here also, there will be one strand like this. Now since you are putting in N14 medium, obviously this will again have like this only. Okay, so this and this will be obviously hybrid always. So 2 of them will be always hybrid, rest all of them will be 14, 14, 14, 14. So out of 8, how many? 2 of them will be hybrid, rest all will be normal ones. Okay, N14, N14. It goes on like this, and that's how the question comes as well. Now if you are asked for 80 minutes, how will you find out? If you are asked about 80 minutes, which is given in your textbook, there is a question like that. What will happen after 80 minutes? So in 80 minutes, how many times will the replication process take place? It just takes 20 minutes for one replication. So 80 means 4 rounds, 20 plus 20 plus 20 plus 20. 4 rounds, right? So 2 is to the power 4. What is it, children? 16. Right? 2 is to the power 4, 16. 2 into 2, 4, 4 into 2, 8, 8 into 2, 16. There will be 16 of them. Now out of 16, how many of them will be hybrid? 2 of them will be hybrid, rest all of them will be 14, 14, 14, 14, 14. Okay. So he was able to find that basically this experiment showed that whatever you do, there will be copies of things being made, and it will be incorporating the ones that is the isotope that is put in the medium, and it is showing semi-conservative mode, that which is why you still find, where is it? Which is why you still find this thing. Otherwise you would not have found, right? The parental variety is still there. The parental variety is still there everywhere, which is helping in the formation of the new one. Okay. If parental variety was not copied, it was not used for replication, then probably this would have been discarded. But it's not happening like that. So I don't know why is this here. Okay. Now let's read this once. So then they transferred the cells into a medium. Okay. So first they put it in N15. Okay. You know that 15NH4Cl medium. When they put it, all of them had this DNA, right? And when they put in the cesium chloride band, they saw the lowest band, which is the heaviest one. Okay. Now what they did is that then they transfer the cells into a medium with normal NH4Cl, but with 14 isotope, okay, and took samples at various definite time intervals as the cells multiplied, and extracted the DNA that remained as double-stranded helices. The various samples were separated independently on CSCl gradient, that is cesium chloride gradients, to measure the densities of DNA. Thus, the DNA that was extracted from the culture one generation after the transfer from 15N to 14N medium, that is after 20 minutes, E. coli, okay, had a hybrid or intermediate density. So over here, if you see, they had a hybrid or intermediate density. I am writing intermediate as well. Intermediate density. Okay. Now, where is it? DNA extracted from the culture after another generation, that is after 40 minutes, was composed of equal amounts of this hybrid DNA and that of light DNA. So in the next generation you saw that 2 of them were hybrid and 2 of them were normal. So it was equal. Okay, that was the thing that they said. Apart from that, there is nothing that they said, and they asked you to find it. Okay, they asked you to find it after 80 minutes also. That is also given in your NCERT. So with this we come to know that the semi-conservative theory of replication is actually true. It's actually true. It is keeping the parental variety and making a new one. This was done by Meselson and Stahl on E. coli. The replication time of E. coli is just 20 minutes. Please remember that. It's a very important piece of information. Okay. Works? Oh, I had one box. Okay, now very similar experiments involving use of radioactive thymidine to detect distribution of newly synthesized DNA in the chromosome was performed on Vishya faba, that is faba beans, okay, by Taylor and colleagues in the year 1958. This is a PYQ that has come. The experiments proved that the DNA in chromosome also replicates semi-conservatively. Semi-conservation means half of the chromosome, half of DNA is still from your parent. Okay, the parental type of DNA is also there. That is semi-conservation. Fine. So there was another experiment done on Vishya faba, which is faba beans. It's a type of beans. Please remember the name. And it was done by Taylor and his colleagues in the year 1958. Okay, in the year 1958 it was done. Here it was 1958, same thing, same year. So over here also it's the same year where things were done. And over here, what did they use? They used radioactive thymidine. There they didn't use radioactive things, okay, they were using isotopes which would differentiate on the basis of density, of their density. N15 had heavy weight, whereas when you talk about N14 it was lightweighted. Cool? Will you be able to remember this? Okay. Now, first question, try to do this and tell me in the comment section. So, Escherichia coli fully labeled with 15N is allowed to grow in 14N medium. Okay, the two strands of DNA molecule of the first generation bacteria have what? Different density and do not resemble parent DNA. Different density but resemble parent DNA. Same density and resemble parent DNA. Same density but do not resemble parent DNA. You have to try it out by yourself and do let me know. Okay, here now itself. Pause the video and do the question. Next question for you, which one of the following is not applicable to RNA? Heterocyclic nitrogenous bases, Chargaff's rule, complementary base pairing, 5-phosphoryl and 3-hydroxyl ends. Okay, do it now. Third, semi-conservative replication of DNA was first demonstrated in Escherichia coli, Streptococcus pneumoniae, Salmonella, Typhimurium, Drosophila melanogaster. We did it now. Okay. Fourth one, a molecule that can act as a genetic material must fulfill the traits given below, except it should be able to express itself in the form of Mendelian characters, it should be able to generate its replica, it should be unstable structurally and chemically, it should provide the scope for slow changes that are required for evolution. Easy question, no? This is very easy, you can write out. Going ahead, the pneumococcus experiment proves that bacteria do not reproduce sexually. RNA sometimes control the production of DNA and proteins. Pneumococcus was not done here. In the previous session I spoke about it, so you need to know. Bacteria do not reproduce sexually. RNA sometimes control the production of DNA and proteins. DNA is the genetic material and bacteria undergo binary fission. What is the correct answer? Try to tell me. So, let me solve all the questions that I gave you, okay? And you check if your answers are correct. Escherichia coli fully labeled with N15. So, I understand that it has two strands which has 15N, okay? Is allowed to grow in N14 medium. It is allowed to grow in what, children? 14N medium. Fine. The two strands of DNA molecule of the first generation bacteria will have. So, first generation bacteria as in when I am putting this over here, okay, these two strands open up and N14 is there. So, this comes, now I have two hybrid, okay, and so different density and do not resemble the parent DNA. Do they resemble the parent DNA? No. Do they have different density from the parent DNA? Yes, because 15-15 and then it will be in the down region, but if it's hybrid it will be in the intermediate density. So, they will have children different density and do not resemble the parent DNA as well. Okay. Now, which one of the following is not applicable to RNA? Heterocyclic nitrogenous base, Chargaff's rule, complementary base pairing. I told you it's Chargaff's rule because it can be single stranded as well at times, and at that time you cannot expect them to follow the Chargaff rule, which states that if A is this much, adenine is this much, the thymine also will be this much only. It will be always equal. No, it cannot be possible if it's a single stranded DNA. Sorry, if it's a single stranded RNA. In this case, single stranded, so it can be A, there can be more amount of T, there can be more amount of A again, anything can happen, right? So, I cannot claim that A and T will be same. If it had a complementary pairing, 10 obviously. So that is lost. Now semi-conservative replication of DNA was first demonstrated in, it was done by Meselson and Stahl. It was demonstrated in Escherichia coli, which has a replication time of about 20 minutes. Cool, my little kids. Going ahead, a molecule that can act as a genetic material must fulfill the traits given below, except it should be able to express itself in the form of Mendelian characters. Yes, children. It should be able to generate its replica. It should be unstable. No, children. It should be stable. No, that's why RNA was removed. So this is the correct answer. This one is the correct answer. Okay, because you are asking must fulfill, except. Okay, so apart from this, everything should be fulfilled. The pneumococcus experiment, okay, where Streptococcus pneumonia was used, it was done by Griffith, okay, where he was putting R strain and S strain, the virulent strain and non-virulent strain in the mice, and seeing what he's understood that something is getting transformed to something else, which is why the normal strain got also virulent. So in there, DNA as a genetic material was discovered. So this is the answer. This is not the answer. Okay, got it? So these are the 5 things. If you were not able to do the last question, please do not worry. You can refer to NCERT by yourself, or you can watch the video that I made for you. It's for you children, it's not for me. Obviously, anytime it's available in your this thing, what's say, playlist, right? So anytime you can go and watch this. Now one question I have for you, are you, okay, oh, so one question is not there today because now we are going to celebrate Dussehra, right? So I'm wishing you a happy Dussehra. So for 2 days you won't be having any classes. You can enjoy your time. We would be making a separate thing for it as well. But as of now, I'm just colloquially telling you, you can enjoy your time. Or if you think that, okay, enjoyment has been done, now I want to study, you can also study for it. Okay, study all the chapters that we completed in biology and then give the mock test, which is on Sunday. So Sunday's mock test, obviously we cannot postpone it, do anything with it. So Sunday we will be having mock test, that is for sure. And in this mock test, I'll tell you what all is coming. So I have given 20 questions from this chapter itself, but 10 questions will be from all the chapters, other chapters that we have studied. Okay, so in total we have 30 questions. I request you to try out mock test. It has all the previous year questions. There's nothing that I made up. Okay, so previous year question practice is very much important. So we thought in the form of mock test only, we'll have a separate timer for it and try to solve things. Okay, and I wish you a very happy Dussehra, my little champions, and I hope you are happy, right? So that's all. If you think that this particular video was helpful for you, then do not forget to subscribe to our channel, and also do let me know your genuine comments regarding this. I'll be really happy to read all of them. Yeah, so bye bye children, have a blessed weekend. Bye bye, take care.